Time and again, we've said, patch clamp measured it. But this instrument, the gold standard of electrophysiology, how does it actually work? On a raw recording, what do those winding curves really say? Today, we go inside and learn to read a current trace from scratch. Understand it, and you understand the very first instant behind all the data we've shown. First, the core. A glass microelectrode, its tip only a micron wide, gently touches the cell membrane, and with a little suction forms a seal with a resistance of over a gigaohm, a gigaseal. That seal electrically isolates the tiny patch of membrane under the tip from the outside world. From then on, every ion the channels let through cannot escape the electrode's eye. So how do you measure? With a trick called voltage clamp. It forces the cell's membrane voltage to a value you set, holding it fixed. Then it measures how much current the instrument must inject to pin the voltage there. And that current is exactly equal to the ionic current the channels are passing at that moment. Whatever voltage you apply, whichever instant you watch, the channel's response is laid bare. Let's read a real recording using hERG. First, give the cell a depolarizing voltage step to open the hERG channels, then snap the voltage back to a negative value. In that instant, the channels haven't closed yet, potassium gushes outward, and the curve draws a signature hump, the hERG tail current. How tall that tail is tells you how active hERG is. Now watch the magic. Add the test drug outside the cell, and as the concentration climbs step by step, you see that tail current press down bit by bit. Plot the percent block at each concentration as a point, connect them, and you get a beautiful S-shaped curve, the dose-response curve. And the very middle of that curve, the concentration that halves the current, is the IC50 we've discussed from episode 1 to today. In practice, there are two styles. Automated patch clamp measures hundreds of cells at once, fast for high-throughput screening. Manual patch clamp goes cell by cell, slow but precise for gold standard confirmation. But whichever it is, a trustworthy recording has hard criteria. The seal tight enough, the baseline stable, leak current subtracted, and the result reproducible. These unseen details are the real backbone of a testing report. So you see, from a glass electrode touching a single cell, to a tail current to an IC50, to the virtual heart's net charge, to a report the regulators accept, this whole interlocking chain, its lowest and most humble act, is patch clamp hearing the voice of a single ion channel with its own ears. That is why this decades-old technique remains, to this day, the irreplaceable gold standard for cardiac safety and for ion channel drug development as a whole. With that, Season 1 has just one episode left. We distill this entire cardiac safety story into a single map you can take with you. Follow Channel Observatory, and we'll take them one channel at a time.